Okay. Hello, everyone. My name is Jay Sung from the Mayo Clinic in Rochester, Minnesota. Today, I'll be presenting some of the latest work from my research group in understanding more about what the microbiome can tell us about health. Before I begin, I'd like to mention that the work I'll be presenting today has recently been published. This study was led by a terrific postdoc in my group, Dr. Vinod Gupta. So for those wanting to know the full details of our work, please either read the paper or feel free to shoot me or Vinod an email. As many in this audience will know, the gut microbiome has a strong influence on almost every aspect of human biology. Here on the left, I provided a few examples where the gut microbiome has been shown to play a significant role. Now it's assumed that when these pathways and modules go wrong due to something in the normal gut microbiome being perturbed, then the result can lead to an assortment of diseases, such as the ones listed on the right. Now, although many of the gut microbiomes of these diseases have been studied individually against healthy controls, the question of what a gut microbiome that encompasses all diseases could look like, and thereby basically investigating the non-healthy gut microbiome, has not been widely considered despite the profound significance. Seemingly related to this topic, an interesting meta-analysis came out from Eric Alm's lab at MIT back in 2017, where they found that gut microbiome signatures can be shared across multiple studies and even multiple diseases. First, I'm showing on the left 3 heat maps, each corresponding to a particular disease, where rows are bacteria, columns are independent studies investigating the respective disease, in this case, obesity, colorectal cancer, and CDI, or Clostridium difficile infection. The darker the red means that the bacteria was higher in disease, and the darker the green means the bacteria was higher in controls. As you can see by comparing across columns, bacteria was either high or low in disease consistently across studies, showing some reasonable amount of reproducibility in disease-associated gut microbiome signatures. What was really interesting is what the authors found next. Bacteria whose abundance patterns were commonly observed across multiple diseases. On the heat map on the right, the columns are now bacteria and rows with the bars indicate whether the bacteria was seen across 2 or more diseases, hence non-specific to a disease, or unique to a single disease. As you can see on the top row, there are many bacteria that show consistent patterns, either up or down in disease across multiple diseases. Towards the bottom, you can see gut microbiome signatures found in only one disease. So there are indeed several bacteria that are disease specific. Now, that gut microbiome signatures can be shared across multiple pathologies led us to wonder the following. Can we identify gut microbiome signatures of disease irrespective of the clinical diagnosis and use this signature to design a stool-based test that predicts health, or in our study, predicts how likely you are to have a disease or not? If successfully demonstrated, this idea could be useful in the following. Say a patient walks into the clinic for a routine checkup, gets his normal blood work done to look at fasting blood glucose levels, cholesterol, etc., and then integrated into this checkup would be a gut microbiome test, showing you not only what species or strains are up or down, but also any signs of something being potentially off. Actually, if you talk to clinicians, at least where I work, they get asked all the time by their patients on how they can learn more about their gut microbiome and how it relates to overall health. So definitely a niche for this type of work exists. Okay, but where do we start? This is a simple infographic I bring up with my colleagues who aren't familiar with the details of the biomarker discovery or the molecular signature discovery work our group does. Oftentimes, people think that analyzing omics data entails a bunch of coding and data parsing. Although there are some elements of truth to this, rather, the majority of the time is actually spent on getting good quality training data or the discovery cohort necessary for the machine, algorithm, software to learn something new and useful. Afterwards of getting this training data, or essentially the search space we'll be working with, we can then focus our efforts on the experimenting and coding, which will involve feature selection, model selection, and model optimization and parameterization. So first, let me explain how we obtain the training data. Fortunately, All the data we need is actually available in the public space. Shown here are details on our multi-study gut microbiome data integration pipeline. But due to time constraints, I'd like to quickly mention that we started from a collection of over 7,500 stool metagenome samples pulled from 55 independent publications. And after excluding some studies and samples using a strict criteria, the final training data was a pooled metadata set of 4,347 stool metagenome or gut microbiome samples from 34 independently published studies ranging across 13 phenotypes. As shown in the table, these 13 phenotypes include 2,636 samples from healthy subjects. Here, healthy was defined as self-reported to not having a disease or disease-related symptoms. Also, there are 1,711 samples from non-healthy subjects. Here, non-healthy subjects were actually patients with one or of 12 different disease or abnormal body weight conditions. These 12 non-healthy phenotypes include cardiovascular disease, colon cancer, Crohn's disease, ulcerative colitis, obesity, and type 2 diabetes. Now, across all of these samples, we found 785 microbial species observed to be present in at least one sample. Of note, our entire analysis was performed at the species level to get to the finest level of taxonomic precision while still maintaining a reasonable amount of comprehensivity. Okay, so that's a little bit about the training data we will use to develop our gut microbiome health indicator. Next is to find which species are associated with health and which species are associated with disease. To explain how we'll do so, I'd like to use the following analogy. On one side of the screen, you have an environment that is teeming with life, fresh air, good overall ecosystem, but on the other, you have an environment full of waste, toxicity, and definitely not where you want to go hiking or swimming. Obviously, the healthy environment is the one on the left. If you think about it, in the good ecological environment, you have life forms that are much more prevalent than in the bad environment on the right. But one important point that should not be overlooked is that the bad ecosystem on the right is not void of life, but on the contrary, there are definitely life forms in the bad environment that are much more abundant compared to the good environment, or in other words, scarcely seen in the good environment relative to the toxic environment. Okay, so if we were to quantify this concept and adopt it for our own purposes, we can term the microbial species that are more frequently observed in healthy subjects as health prevalent species. You can also term the microbial species that are less frequently observed in healthy as health scarce species. So these health prevalent species can be assumed to be associated with the healthy state, and these health scarce species can be assumed to be associated with the non-healthy state. In the table at the top, we show the top 5 healthy associated species rank ordered based on how more often they're found in healthy relative to non-healthy. Some of you may recognize here the Bifidobacterium adolescentis, which is a known probiotic. In the bottom table, we show the top 5 non-healthy associated species rank ordered based on how more often they're found in non-healthy relative to healthy. You can see that at the top, these are mostly Clostridia and Ruminococcus species. So in summary, we have a prevalence-based strategy to identify species associated with a phenotype, which is basically about taking into account the frequency of a signal. Importantly, the identification of these healthy and non-healthy associated species, or the species prevalent and scarce and healthy, was found using a top-down strategy purely from the training data and not a bottom-up approach wherein we first assume something to be there and then try going after it. But then the next question is, how do we use these health prevalent and health scarce species to predict health or disease? Now, we actually have multiple pages in our paper explaining how we derived and implemented a mathematical formula to predict health and disease, but due to time, I'll briefly describe the main idea in the following. In our view, the most intuitive and straightforward way to estimate whether a microbiome sample is either healthy or non-healthy is to simply find the ratio of the collective abundance of health prevalent species to the collective abundance of health scarce species. And if we put a log on this ratio And here's the really important point, then any output that is positive means higher collective abundance of health prevalent species, meaning healthy. But on the other hand, any output that is negative means higher collective abundance of health scarce species, meaning non-healthy. Now, this collective abundance psi is basically a slightly modified version of the geometric mean, which is a reasonable metric to find expected values in microbiome data, given that the relative abundances span several orders of magnitude. After doing the math, you can find that this version of the mean takes into account the presence, absence of each species within the group, the cumulative relative abundance, and the diversity of those species. Now, moving forward, based on whatever prevalence cutoffs you select, you can have many combinations of or sets of health prevalent and health scarce species. Next, after identifying systematically all possible species sets, we plug each of them into our formula to test model accuracy. Here, accuracy is basically how accurately we predict healthy subjects to be healthy and non-healthy subjects to be non-healthy. And after looking at all the accuracies, the best set of features, that is the two sets of microbial species and the parameter tuned model, is what we use as the gut microbiome health index, which I'll be calling GMHI for short for the rest of this presentation. Here's a table from the main manuscript showing the 50 microbial species we found to best separate healthy from non-healthy. 7 of these species, here at the top, are the health prevalent, and the remaining 43 are the health scarce species. Now, I know the text in the table is very small, but I'd like to point out that some of the health prevalent species, especially the bifidobacteriums, here at the top are actually marketed probiotics, providing some evidence that these probiotics out there in the market are indeed associated with health. Okay, so I just went over how we obtained the training data, how we identify microbes at the species level associated with healthy and non-healthy based on frequencies, and how we designed the mathematical formula that can be used as a classifier to distinguish a microbiome sample as either healthy, which is no disease, or non-healthy, which is having a disease. So what do our results look like? First, we differentiate the two major groups, which are healthy group and the non-healthy group. In the violin plot here, we're showing the GMHI distributions of both groups, where GMHI scores are shown on the y-axis. As I previously mentioned, a positive GMHI means predicted as healthy, and a negative GMHI means predicted as non-healthy. In this way, we get a balanced accuracy of 69.9%, or basically 70%. This balanced accuracy is calculated by taking the proportion of healthy samples we classify correctly and the proportion of non-healthy samples we classify correctly, and just take the average. The best you can do is 100%, the worst you can do is 0%. The statistical significance of the difference between both distributions is an insanely low p-value, and the effect size of this separation, which we use Cliff's delta to calculate, is 0.56. Briefly, Cliff's delta is a rank-based non-parametric procedure, which goes well with the Mann-Whitney U-test. Having a magnitude closer to 1 generally means a better, more consistent separation between the two groups. In addition, we broke down the non-healthy group into its 12 subclasses. Healthy is still in here, but just crunched on the sides a bit. The stars on the top of each of the 12 non-healthy subclasses indicate a significant difference with the corresponding phenotype to healthy. Here, 11 of 12 show significant separation compared with healthy. But to truly appreciate and utilize GMHI, we need to consider it as a continuous variable rather than considering it as black and white absolutes. Let me explain. In the plot in the middle of the screen, we're binning all 4,347 samples into bins of GMHI. On the left and right, you have the two extremes. In one bin, you have people whose GMHIs are from +4 to +6. And in the bin on the other side, you have people whose GMHIs are from -4 to -6. In between these bins, you have samples binned in a stepwise fashion leading to both ends. The points on the blue line here indicate proportions of samples of each bin that are actually healthy. points on the orange line indicate proportion of samples of each bin that are actually non-healthy. The take-home message here is that as your GMHI is more positive or more negative, the higher the confidence we have in the prediction accuracy. The closer your GMHI is to 0, which indicates an equal balance of health prevalent and health scarce species, the less confidence we have in whether you have a disease or not. Perhaps this point can be explained further using ordination plots. Here, when we plot all 4,347 samples onto a principal coordinate analysis plot, we see that healthy and non-healthy samples merge onto each other, showing not so good separation. But what if we only look at the most healthy samples of the healthy group and the most non-healthy samples of the non-healthy group? We see that if we look at the top 10, 25, 50, and all the way up to the top 100 most healthy and non-healthy samples based upon their GMHIs, we actually get pretty good separation. This shows that GMHI is actually a decent way to stratify healthy and non-healthy groups if we consider the actual index values rather than looking at only positive and negative. But how does GMHI do compared to other metrics? Well, this is not very straightforward to answer, as there is no other gut microbiome-based metric specifically designed to tell you about health. So just for comparison purposes, we just use what's commonly used in the ecology literature, mainly Shannon diversity, species richness, and 80% abundance coverage, which is basically the minimum number of species you need to compose at least 80% of the total abundance of microbes in a sample. I like to direct your focus to the cliff deltas or the effect sizes I mentioned earlier. As you can see from the small letter Ds in the 4 violin plots, GMHI has the highest magnitude, where again, being closer to 1 indicates the stronger separation between healthy and non-healthy. On each of the scatter plots, we compare GMHI head-to-head with each ecological metric. Each point corresponds to a microbiome sample. The x-axis plots each ecological metric, and the y-axis plots GMHI. If you compare the distribution on the x-axis to that on the y-axis, you'll see that the ones on the y-axis, which corresponds to GMHI, always separates healthy from non-healthy better than every other parameter. So up to now, I hope I've convinced you that GMHI can separate healthy from non-healthy, and much better so than other indices. But really, the gold standard of any biomarker or health disease indicator is how well it performs on an independent validation set, that is, a data set not previously seen during the model training process. Here we show our GMHI results on 679 microbiome samples from 9 additional studies. On the left here, we show the results when asked to classify between the 2 groups of healthy and non-healthy. As before, the blue and orange distributions are for healthy and non-healthy respectively, and the y-axis shows the GMHI scores. This time, we get a balanced accuracy of 73.7% and a Cliffs delta of 0.64. This was really encouraging, as we actually did better here than when GMHI was tested on the training set, where accuracy was 69.9% and Cliffs delta was 0.56. This shows that the overall reproducibility and robustness of GMHI is quite good. Now on the right, we show the GMHI distributions for each class of each new study. As you can see, the 3 cohorts shown in blue, 3 healthy cohorts shown in blue, all score mostly positive distributions, and the majority of the non-blue or non-healthy subclasses score mostly negative distributions. Going from left to right, we're showing the GMHI distributions for ankylosing spondylitis, which is a chronic inflammatory condition of the spine, colorectal adenoma, 3 cohorts of colorectal cancer, Crohn's disease, liver cirrhosis, non-alcoholic fatty liver disease, and rheumatoid arthritis. What's really interesting here is that liver cirrhosis and NAFLD, both diseases of the liver, were not seen in the previous training process with 4,300 plus samples, showing that perhaps GMHI can likely be extended beyond the diseases it was trained upon. This is really cool because now we can begin to wonder whether we captured elements of a universal gut microbiome disease signature. Next, we show how well the other metrics, Shannon diversity, 80% abundance coverage, and richness do on the validation datasets. The main message here is that we explain this carefully in our manuscript. GMHI outperforms other metrics by a really wide margin in regards to both accuracy and consistency. So in summary, we designed a stool-based test that looks at your microbiome to predict the likelihood of having a disease. This is really important because if you think about it, cancer, type 2 diabetes, metabolic syndromes do not happen overnight. Rather, these diseases take years or even decades to develop inside yourself. If we can look at your gut microbiome and detect something off about you early or prior to the onset of major symptoms, which is when you're really in trouble since it becomes much harder to reverse well-progressed disease course, do we then have a potential tool to screen for the pre-disease state? To develop our index, we used shotgun stool metagenomes pulled from independent studies. GMHI is basically a log ratio formula, balancing the collective abundances of two sets of species. One set of species found more frequently in healthy compared to non-healthy, and another set of species found less frequently in healthy than in non-healthy. GMHI outperforms methods adopted from ecological principles in both training and validation datasets. And we believe our study paves the path forward to track, understand, and predict human health through non-invasive approaches. So to bring everything to full circle, I'd like to reintroduce the clinical setting or consumer scenario in which this tool may provide the best utility, because at the end of the day, we want to be users of our own tools. So on top of a report that provides the high-level taxonomies you have, or ordination plots describing beta diversities, or in addition to a report on alpha diversity, which I've shown above that it doesn't really mean much, or on so-called bad species, which any infectious disease or GI doctor can tell you that you should not be having in the first place, or to complement reports on functions your microbiome has or does not have, or rather than providing some ambiguous score that no consumer will ever know how it was conceived. I imagine having a score or index which you know exactly how it was developed and what it's measuring, that you can track at any moment in time, gives you some intuition of whether you have a disease independent of the clinical diagnosis, allows you to compare yourself to others in the population, and even track your GMHI over time to see how well you respond after a dietary or other lifestyle intervention. Plus, it's kind of fun to quantify and monitor things about yourself. So I hope you enjoyed this presentation, and in case there is anyone in the audience interested in working together to take this tool to the next step, say incorporating GMHI into current microbiome reports or consumer products, or making a 2.0 version with more samples, diseases, ethnicities, races, or even a 16S RNA gene amplicon version, or moving this type of work into specific contexts, such as a test for colon cancer, a specific autoimmune disorder, or even trying this out on any type of intervention study or clinical trial, please send me an email and let's discuss. Before I end, I'd like to acknowledge my fantastic research group at Mayo Clinic, especially Vinod here in the middle, who led this amazing work, and also my generous funding sources, without whom this work would most likely not have come to fruition. Now, by going back to our take-home message slide, I'll stop here and take any questions. Thank you for your attention.